The overall goal of the following experiment is to incorporate voltage-gated ion channels into giant unilamellar vesicles and characterize their activity with patch clamp measurements. This is achieved by first partially dehydrating a solution of small vesicles containing the ion channel KvAP to create a lipid protein film. Next, the lipid film is rehydrated, which causes the formation and growth of cell-sized giant unilamellar vesicles, or GUVs, containing the channel. Then, the GUVs are transferred to a recording chamber, and a piece of GUV membrane is excised with a patch pipette in order to measure ion currents. The results show that the ion channels in the GUV membrane activate in response to changes in membrane voltage, based on the analysis of the current recordings.
Giant unilamellar vesicles can help answer key questions in the field of biophysics related to membrane-protein interactions. For instance, the interplay of transmembrane proteins with the physical properties of the lipid membrane.
Though this method can provide insight into the function of the voltage-gated ion channel KvAP, it can also be applied to other ion channels, transporters and pumps, and transmembrane proteins in general.
Visual demonstration of this method is critical as the steps involving the manipulation of giant vesicles are hard to pick up just by reading articles.
After preparing solutions and small unilamellar vesicles containing KvAP according to the text protocol, prepare the electroformation chamber by first inserting the wires through the hole.